Hi everyone, welcome to the first episode of the tutorials on gene expression analysis. For this tutorial, we are going to analyze an RNA Seq dataset and identify genes that are differentially expressed between two conditions. I will talk more about the conditions later on. For this tutorial, we are going to use this workflow which is being displayed, and I will leave the link to the paper so that you read more about this workflow as well. If you are a beginner, I will also encourage you to watch my other video where I give a general overview of RNA Seq data analysis. So please do that. In order for you to follow this tutorial, you need to make sure you have a Unix-like operating system. So with this, I'm referring to Linux or Mac OS. Any of them should be fine. In addition, you should have Anaconda installed on that PC. Instructions for installing Anaconda is in the description box, so please watch those videos to set up your PC. And so without further ado, let's start the practicals. Okay, so we are on the terminal and ready to start the exercise. The first thing we have to do is to get the master scripts. We are going to use the script to download all the necessary scripts we will use for this exercise. This script can be found on my GitHub repo, which is called transcriptomics, and this is the page. I will leave the link to this page in the description box. So here, search for a file called getscript.sh, this file here, and then just get it. So click on it to download, and then you'll be sent to this page. Just click on raw, and then you'll be sent to this page, and here just right click it, and then just go to save pages, and then you can just save it on your file. On the terminal, we can also download it. So to download on the terminal, which I'm going to do, I need to first get a link. So just copy this link here that I am hovering my cursor over, and then use that. So let's go to the terminal, and then I'll show you what to do. So on the terminal, let's first create a directory where we will work in. So we say make the RNA Seq, and then let's cd to it. So now we are ready to download the master scripts. So to download, I'll use wget. So I'll say wget, and then I'll supply it with the link to the file. You can also use curl to download, but the choice depends on you, so that's it. So once we have this, we execute the command. Okay, so the script has been downloaded for us. So let's clear the screen and just check it out. So do an ls and you will find a file there. Because this is a bash script, we have to add execution rights. And so we say chmod +x, and we call the script here, this file. So let's execute it. It's done. So do an ls, and then depending on your environment, the color of the file would have changed. But the color doesn't matter anyway. So far as we have executed the command, that's fine. So we are going to run this script to download all the other scripts. So we say // and then we specify the scripts here to download the rest of the files. Okay, so download is being done for us. Perfect. So all the scripts that we need have been downloaded. So let's clear the screen and check them out. So do an ls and you'll find them there. I will explain what each script or file does as we move along. Because some of them are bash scripts, we need to add execution rights. And so again, we are going to issue the chmod again +x. And this time you will say star.sh. So execution rights will be added to all the files that end with .sh. And so let's execute this command. Perfect, it's done. So do an ls and you will see that the colors have changed again. And so we are ready to roll. The next thing we have to do is to install the tools and packages we need for this exercise. We are going to use Anaconda. And so for this particular installation process, we are going to use the environment file, which is here. So this file here has the details we will need for this exercise. So let's do a cut first to display. And then you see the list of tools, the environment, and channels. But I have a separate tutorial that goes into details with installing tools with Anaconda. So if you want to understand this particular file here, you can just watch that video to get it. The link to that video is in the description box. Now let's proceed. So let's clear the screen and continue. Before we use this environment file to install the tools, we need to add channels. We need to add Conda channels. We are going to use 3 channels here: R, Bioconda, and then Conda Forge. So let's add them. So to add channels, we say Conda config add channels. Let's add the first one, which is R. Let's add a second channel, Conda config add channels Bioconda. Let's add a third one. The same command, but this time we add Conda Forge. Perfect. So now we have all the channels added. So we are ready to roll. Okay, so now that we have the channels added, we are now going to execute another Conda command to install the tools. So we say Conda env create -f, and we specify the environment 1.yaml file, and then we execute this command. And so Conda will check the contents and then use that to create an environment and install the tools for us. So let's wait whilst that is being done. Okay, so installation is complete. Let's clear the screen and continue. The next thing we have to do is to activate our environment. We are going to activate, and then we are also going to install one last package, which is an R package. So let's do that. Now to activate, you need to call Conda and then also specify the environment. And so if you don't know the environment you have, you can just check it out by saying Conda env list. So this will show the environment that you have on your system. Our environment is called RNA Seq. You can also check it here. Let's do a head on the environment file, and you will see it here. This is the name. So it is this name that we are going to use to activate our environment. So let's continue. Let's clear the screen and continue. So let's say Conda activate RNA Seq. Perfect. We now have the environment activated. We are now going to install 2 additional R packages. These packages, I was not able to install them using Conda, so I'll use a different approach. So let's first activate the R environment. So just type R, uppercase R, and then go to the R environment. Let's first clear the screen here. In R, to clear the screen, we use the Control and then the L. So let's do that to get a new screen. One of the packages is available on GitHub, so we need to use another library to help us do the installation. So I'll load that library first. So I'll say library DevTools. Okay, so this library will help us to install one of the packages. So let's install the first package, which is in GitHub, which is hosted on GitHub. And so to install a package that is hosted on GitHub, we use DevTools. So we say DevTools, and then we give our double colon, and then we say install GitHub. Then we specify the package that we want to install. So let's specify that. So I am using the instructions that has been given for installing that particular package in R. So this is how we do it. So we get our DevTools and then they install GitHub, and then we specify everything else in code. So Alisa Frazi, I think that is the GitHub user. So I think that is the account that is hosting the package. So Alisa Frazy, yeah, that's the name. And then slash, and then we give the package name, which is R Skittle Brewer. Yes, so this package here, this is what we are getting. So let's close the brackets. Let's check that everything is okay here. Okay, so we have the spellings correct. So we can run this to install this particular package. So let's do that. Okay, so that package has been installed. Let's install the second package. That one is repo. So let's clear the screen and then just do that as well. So let's do that. For that one, we are going to use the conventional R command, that is install.packages. So we say install.packages and then we give the name. So I'll put it in quotes, repo, like this, and then I'll hit the enter button to install. So here we are being asked to specify a current repository. So I'll just choose, let's say 80. Yeah, let's use 80. And then let's select it. So after that, installation will proceed. Okay, that package has also been installed. So we are ready to go. So since that package has been successfully installed, we will just quit R. So to quit R, we use the kill key, but I'll clear the screen and then just use a new one so that you can see it better. So let's do this. So let's quit R. So we say kill and then we give our parentheses here and then we hit the enter button. We are being asked to save the workspace image, but I don't need to do that. So I'll say N and then I'll just exit the R environment. So now we have all the packages we need installed. So we are now ready to start the analysis. So let's clear the screen and continue. The next thing to do is to download the example data. The link to that data can be found in this particular paper here. And let me also say that this tutorial that I am doing now is based entirely on this particular paper and then the blog post made by Dave Tang. So it is from these articles that I prepared the materials and all that. So credit goes to them. And so this is the link to the data. So you can just click it or you can just copy it. Because I am on a terminal, I prefer to download it on the terminal. So that is what I'll do. But you can also click and then download it. So I'll download using reget. So I'll say reget and then I'll supply it with the link. And so I'll use this command here. So execute this command and then data will start being downloaded for you. So this is how we download it. So let's wait for the download to complete. Okay, so download has been done. So let's clear the screen and just do an ls and you see the data here. This is a compressed file. And so what we will do is to decompress it. That's what we are going to do. And that means we are going to extract the contents. And so to do that, we will say tar xvfz and then we supply the file name like this. And so once we do it like this, we will then execute the command. So everything will be extracted for us. Okay, extraction has been done. Let's clear the screen and continue. So do an ls again and you see this directory here. So this is what was extracted for us. And that is what we are going to use. So let's check the content. So do an ls into it and you see some files and directories there. You can also use the tree command which might not be necessary for you. It might not be necessary, I repeat. So you can use this to also check the directories in a tree form, and then you will see where each file is located. Okay, so let's proceed. Now we have the directory, and so we need to prepare this particular file. There are a number of files we will use, and there are those we don't need. And so to make it simple, I have prepared a script that we will use to prepare the data. So we will run the script here to prepare the data that we will use for this exercise. So run the script. So say /prepare data and specify the directory, this one here, and then run it. Perfect. So everything has been prepared for us. And so you also have this statement here telling you where you will find your files. So do an ls again, and you see a directory called resources here. So this has all the files we need. Okay, so do an ls again into it, and you see our files there. So we have our reference genome in the FASTA format. We also have the annotation here. We have our metadata here. We have our samples. So let's look at the samples first. So do an ls resources samples, and you see the files here. So these are FASTQ files. These are paired and reads. That is what we are going to use. Let's check again. Let's clear the screen and continue. Let's do an ls into resources again. Okay, so let's proceed. So we have our FASTA file. We have our annotations here. Let's also check this CSV file. This has information that we will use later. So let's do a cat resources and then specify this CSV file. Okay, so this file here has information that we will use. So we have the sample IDs here. We have sex. We have population. So these are the file names, and then we have sex and population. So these are groups. These are all groups. So we have two different groups here. We have sex, we have population. And so that means that the samples here, if you are talking about sex, they will either belong to male or they belong to female. If it's population, they belong to YRI or GBR. RNA Seq experiments are usually case control experiments. And so when we are identifying differentially expressed genes, we are looking at genes that are differentially expressed between groups. And so for this tutorial, our group of interest is sex. That means we are looking at genes that are differentially expressed between males and females. That is what we are going to do here. And so that is what I thought I would let you know before we begin the actual analysis. But of course, you can also experiment with this, but let's just focus on this particular one here. We will go into details with these ones later on. So these are things you also have to do. Whenever you are going to analyze data, you need to explore and understand your data. And these are things you have to do to get that information. So we are ready to roll. So let's start with the analysis. Now let's also check the sample number. If you look at this, you can check the sample number here. So if you do a cut, these are headers. We don't need the headers, but if you do a cut, then you can just do a WC-L to count. You will have 13, but we have 12 samples. The headers here will not be parts. That's what you need to know. Okay, so that's what you can also do. Let's just say ls resources samples. So we have them here, but you can just do an ls samples and say *_1.fastq.gz and also count the WC-L. And you have 12 samples there. So that's another thing you can use to also check the number of samples. So if you want the total number of files, you don't need the WC-L. you can just do it this way, wc-l, and you get 24. But because it's paired end, you divide by 2 and then you get your 12 samples. And so that is what it is. So let's proceed. We are now ready to start the analysis itself. So let's clear the screen and continue. The next thing we are going to do is to index our reference genome. We need this information because indexing helps programs to do a faster lookup of the genome. And so this is like a preparatory step for the next stage, which will be mapping. And so let's proceed with the indexing. So because we are also dealing with a human genome, I think I forgot to make mention of it. We are dealing with a human genome, and because of that, we also need to get information about the splice sites and then exons. And that is why this indexing is also very important. So let's first create a directory called index. And then let's get the splice sites and exon information into that directory. So let's start with the splice sites. So we say extract splice sites .py, and then we indicate or we specify the annotation. So it's located in the resources directory. So that's this one here. And we specify the output file. So the output will go to the index directory, and we say index/chrx.ss. So this is the splice site information. Let's also get the exon information. So we say extract exons.py. We specify the annotation again. The output file will be index/chrx.exon. So we have that there. Let's do an ls in the index directory. So we have the files there. Now we are going to cd to this index directory first. And let me say that all these things are done using HISAT2. That is the tool we are using for this particular work. So as part of the HISAT2 software packages, we have these Python scripts that do the extraction for us. Okay, so that's what we have here. Now that we are in the index directory, we will continue to do the indexing. We are going to now build the index. Of course, you can also explore. Let's just do an exploration first. Let's check the contents in these two files. So let's just do a head-n, let's say 2. Let's check the exon. We have the information there. Let's also check the splice sites. Okay, so we have some information there as well. So cool. Okay, now we are going to build the index. And so we are going to call HISAT2 build to do that. So we say HISAT2 build. And we specify the number of processors. So we say -p, and then we specify 8. Now, if you have lots of computing power, if let's say you are running on an HPC server where you have a lot of cores, you can increase this number here to a higher one. Let's say 10 or 20 or whatever. So that will make HISAT2 work faster. But this particular number here should be good for a normal laptop or a desktop PC. So that is why I'm keeping it this way. Now let's proceed. So we have this information. So we now have to specify the splice site. So we say --s, and then we specify chr. That's the file that we save the splice site information. So that is chrx.ss. We also need to specify the exon. So let's give the file name. And then we also need to specify the reference genome, the FASTA formats. That one is located in the resources directory. So it's /resources/chrx.fa. That is the name. We are using // because it's located in a previous directory, in a parent directory. So that is why we have it this way. Let's also specify a name. There's a prefix. So this name will be given to all the files that will be generated after the indexing has been done. So let's say CHRX_TRAN. That will be the prefix. So this name will be given to all the files that will be generated. Perfect. So now we will execute this command and build the index. Okay, so indexing has started. So let's wait for it to complete. Okay, so indexing has been done successfully for us. Let's clear the screen and continue. Okay, so if you do an ls now, you see that we have some files created in addition to the exon and then splice site files. So these are the files we will use later on. So now let's cd back to our previous directory and then proceed. The next thing we are going to do is to map the reads to the reference genome. We are going to use HISAT2. So what HISAT2 will do is that it will pick the reads, the FASTQ reads we have, and then identify locations in the reference genome where these reads best matches. And then that will be used to infer transcripts that are expressed. So that is what we are going to do. So mapping in general helps us to identify transcripts, okay, or express transcripts or genes, and then also gets the information about their location. So that's it. So I have prepared a script for doing the mapping. So let's just do an ls and look at the script. So you see the script here called mapped read. So it has the commands we use. So let's do a head. Let's say head n5. Yeah, so let's just increase the number to get the entire, let's just say 10. Yeah, so we have some commands here. So basically, we are going to supply it with the samples. Okay, I think that's also done here for us. So all that it does is to look at the directory that has the FASTQ files, and then it will pick these files and then use HISAT2 to map them. So that is what we are going to do now. So let's proceed. So let's just continue. Let's clear the screen and then proceed. So we are going to execute it. So we will say /map reads.sh, and then mapping will start, and we will wait whilst HISAT2 does the mapping. So let's wait whilst everything is being done for us. Okay, so mapping has been done. Let's clear the screen and continue. So do an ls and you see that another directory has been created. This directory here. So this has the mapped files, let me just say the alignment files. So do an ls into it and you see these files. So these are some files. Okay, that is what we have. And so the next thing we are going to do is to sort these mapped reads. We are going to sort them. And so we are going to sort, and then by sorting, it enables the efficient access of the data. So we are going to sort everything here by chromosomes and contigs as well. And so that's what we are going to do. And so for the sorting, we are going to use SAMTOOls. And in the course of sorting, we are also going to convert the SAM files to BAM files. So SAM files are usually large, and normally what we do is to convert SAM files into BAM files, which are lighter. And so we will do both at the same time. So again, there's a script to do that, and this is called a sort mapped rates. You will use that to do the sorting. You can also check the file out. You can just use a cut. Let me just do it here for us to see. So we have the SAMTOOLS command doing the sorting for us, and then we will get our BAM files. So let's proceed. Let's first run it. Okay, so let me just ls into the map directory again. Yeah, so that's fine. Okay, so now we are going to issue the script to do the sorting for us. So again, I'll issue this command and then I proceed. Okay, so sorting is going to be done for us. So let's wait for it to complete. Okay, sorting has been done. So you can do an ls into the mapped directory again, and we should see the BAM files. So now we have BAM files and then the SAM files. Let's clear the screen and continue. Let's ls again. Okay, so for the downstream analysis, what we need is the BAM file. So we can remove the SAM file to save space. So to remove it, you say rm mapped star.sam. So this will remove all files that end with .sam or .sam. So that's how we do it in Linux. So let's run this command to get it done. So now all the SAM files have been removed. So do an ls into the map directory, and you see that we have only the BAM files in there. So that is it. So we are ready to roll. The next thing we are going to do is to assemble the transcripts. This will enable the accurate quantification of genes and their respective isoforms. We are going to use a tool called STRINGTIE. And again, I have prepared a script already. So let's check the script out. So it's called assemble transcripts, which is here. And so we are going to use it. Let's just do a cut to display the contents in this file. So we are using the STRINGTIE command here to do the assembly. So all the samples here will have their respective transcripts assembled. We also specify the reference annotation, and then we give the output file as well. So just in case you want to know what these commands are, that's it. We are also using 8 processors, so -p will do that. So again, if you have lots of CPUs, you can increase this number here. Now let's continue. And let me say that I'm going to leave the links to documentations for each of these tools in case you want to read about them. And so that is it. So let's do an ls and then let's execute the script to assemble the transcripts. So let's start. Okay, so the transcripts are now being assembled. Okay, so STRINGTIE has been able to do the assembly for us. And so there is a new directory that has been created. Let's do an ls to check it out. So this is the directory. This directory has the assembled transcripts. Let's clear the screen and then proceed. So do an ls into it, and then you see the files there. So that is it. You can even check, let's just do a head. Let's check one of the samples out. Let's use the first one here. Let's just check here. And then we just call the file. Okay, so this is how it is. Okay, so we have the labels, we have the gene IDs and all that. So you can just check, it's just a text file. It's just the extension that has been changed, because with these bioinformatics datasets, they have different formats, and that also is determined because of how we want the information to be represented. So basically, most of these files are text files. So that is what you need to know here. Now let's proceed. Let's clear the screen and continue. Now, the next step we are going to do is to merge all the files that were generated after assembly. Okay, so all those GTF files that were generated, we are going to merge all of them. And so we will still use STRINGTIE to do it, but then we need to first get the file names all into a single list or a single text file, and then we'll continue from there. So let's get the files. So let's say ls assembly star.gtf. We want the file names, and then we put all of them to another file which is called merge list.txt. So this is it. We can just do a cut to get the list here, which is fine anyway. So that's it. And so once we are done with this, we then call STRINGTIE again to merge them. And so to merge, we say STRINGTIE. We specify the merge option -p. Let's use 8 again, and then -g. That is the reference annotation that will be in the resources directory. Yeah, which is here. So chrx.gtf, and then -o, that will be the output file. So let's say STRINGTIE _ merged.gtf. And then we also need to specify the lists, okay, the list of files we want to merge. And this list here is here, merge list.txt. So we need to specify that file. So we say merge, let's do it again, merge list.txt. And then we run it. So everything has been merged for us. So do an ls and you will see a new file, which is here. This has all the transcripts together. So we can even export. So let's just say head. Let's just say head STRINGTIE mesh.gtf just to get the file. So we have basically merged all of them there. Now we can also look at how many transcripts were identified. And so that is in total. Let's clear the screen and check that out. So to find how many transcripts were identified, we will use the cat command. And then we also use grep here, -v. And then we specify this pattern here. And so basically what we are doing is that we are looking for all the lines that do not contain this. That is what the grep-v does. Okay, so it's just an inverse. And then those are the lines we want, but it's not all the contents. We want a specific column. And so we use the awk to get that column here. So that is what we are doing here. So these commands here makes life convenient for you if you are processing text files. So let's continue. So awk.array, and then we add this as well. So transcripts, and then we issue this print command here, and then wc-l. This is it. So this will get the count for us. So let's just cross check to make sure everything is in order. Okay, perfect. Let's check. It seems I omitted something here. Let me go back and check. Yeah, okay, I omitted this one here. Yeah, this one here. So I'll use that to do it. That's also one thing about programming. You need to make sure you put in the right commands. Because I omitted this particular symbol here, I had this error coming up. Okay, this is 0, but we are not expecting 0, so it's more like an error or an unexpected result. So this will give me the correct output. So let's just run this command. Perfect. So now we have the number here. So 3,971 transcripts were identified. So that is what we have here. Okay, so let's continue. Another thing we can do is to compare the list of genes and transcripts that have been assembled, that have been identified. We can compare that to what is present in the reference annotation file. And to do that, we use the tool called GFF compare. So let's just check that also out. So we can say GFF compare -r to specify the reference, which is resources chrx.gtf. And then we also indicate what we have. When I say what we have, the one that was assembled, our data. So we say -g, and then we say -o. So -o indicates that we want the output file to be called merged. And then we specify our data, which is STRINGTIE _merge.gtf. And so we can run this command. Perfect. So everything has been done. So if you do an ls, you'll find a file called merge.stats, which is somewhere here. So this has the statistics. So let's do a cut to get this file .stats. Perfect. And so we have it done. So here we will focus on the transcripts, and then we also focus on these metrics, sensitivity and then precision. So if you look at the sensitivity for transcripts level, it's 99.1. Precision is 52.5. Okay, so that's what we have here. And please note the values may change slightly for you, but I mean we are just using it for practical session, so there might be some slight changes as well. So just take note. Usually some of these softwares, because they are always being updated, it may be possible that the algorithms may change slightly. So some of these values may differ slightly if you are using a different version of STRINGTIE. So please take note of that. Now back to our column. So sensitivity and precision. Now if you look at the paper that has a tutorial. So there's this other one that's for Nature Protocol. If you look at it, it describes the STRINGTIE, the GFF, they describe the GFF outputs, the metrics, so sensitivity and then precision. So sensitivity here, let me just close this. So sensitivity is defined as the proportion of genes from the annotation, that is our reference genome, that are correctly reconstructed. Here, reconstruction will also refer to assembly. Okay, so if you look at our results, we see that there is a high sensitivity. That means that a large proportion of the genes in the reference genome were reconstructed correctly. Okay, that is what this statement means in terms of sensitivity. When we talk about precision here, it refers to the proportion of the outputs that overlaps with the annotation. So the output here refers to our output file, what we have generated after getting our samples assembled. So the proportion of it that overlaps with the annotation. So if you look at precision, let's check back. If you look at precision, we have 52.5% of them. So we have quite a number, okay, quite a significant proportion of this is what is found in the reference. That's what I'll see. And so here, what it also means is that we have quite a significant proportion of our outputs being novel genes or novel transcripts. Let me just use transcript because we are dealing with transcripts. So quite a significant proportion of these transcripts are novel. They are novel because they are not found in the reference annotation. It could also be possible that they are false positives. Okay, so doing this comparison will give you this figure so that you can use that to understand your data and also make decisions. And so from the documentation again, from the paper again, it tells us that this particular measurement may not be able to capture the true accuracy of the assembled genes and transcripts. And so it's important to take note of that. So in my view, you should use GFF compare to try and get some information that will help you to make decisions with regards to your analysis and then the output. That is what I would say here. So just make sure you explore. Usually for some of these tasks, you need to explore, you need to read papers and all that before you be able to come out with or arrive at the answer or the results that you want. So, and that is what this tutorial is about, to give you that introduction so that you can start doing your own stuffs. Okay, perfect. So let's proceed. So we are done with the comparison. Now we also need to do an estimation of the abundance. So this is where the gene quantification is of S. We are now going to quantify the genes and then the transcripts. So let's go back to the terminal. Let's clear the screen and continue. So we are going to estimate the abundance of the transcripts and then the genes. And again, I have the scripts there. Okay, so the script is called estimateabundance.sh. That is it here. So we are going to execute the scripts here to get the abundance for us. So let's execute it. So / and then we say estimate abundance. So it is being done for us. So let's wait whilst it does that. Okay. Again, we are using STRINGTIE. So STRINGTIE will do that also for us. So let's wait whilst abundance is being estimated. Okay, so it has been done for us. Let's check it out. Let's do an ls, and then you see this directory called abundance. So do an ls into it, and you see the individual samples and their respective files. So let's take a look at one of them and see the files. Perfect. So it is these abundances, it is these files that we are going to use to identify the differentially expressed genes. So take note of that. And so that is it. So let's proceed. So from here, what we are going to do is to use R for the rest of the analysis. So R packages are going to be used for identifying the differentially expressed genes. So we are going to use Ballgown and then some other packages to help us do the rest of the analysis. So let's proceed from there. So let's clear the screen and then let's type R. Perfect. So now we are ready to start the exercise. Okay, it is now time to identify the differentially expressed genes. We are using R for this exercise, and therefore we need to load the needed packages. We are using 5 packages here, and so let's load them. So we say library, they should be in lowercase, so library, and then we specify the first one, which is Ballgown. Let's load the second one, which is GenFilter. So library, GenFilter. Let's load the third one. Let's load the fourth one, which is DevTools. Let's load the last one, which is DPLYR. Perfect. So we have all the libraries we need. Let's clear the screen and continue. To clear the screen in R, you use Control and then L. L for laugh, L for light. So just take note of that. Now it's time to load the sample information. So let's do that. So the sample information will help us to get the samples and then the groups that they belong to. So this is required by Ballgown for the analysis. So let's load them. So we say pheno data, and then we read the data. So there's a CSV file, so we use read.csv, this function here, to do that. So what we are doing is to assign the entire operation to this variable. Let's load it and you'll see what I'm talking about. So we have read.csv, and then we specify the path. So geuvadis underscore phenodata.csv. So this is the path. Let's check. Okay, I think we have to give the full directory. It's located in the resource directory. So let me just repeat again here. So resources / GEUBADIS underscore phenodata.csv. So that is what we need. In some tutorials, you see double quotes here and then here. That's also accepted because we are dealing with characters. I also use this tutorial to explain a couple of things to people who may be beginners so that you understand the R syntax. Okay, so let's run this command. Okay, so the information has been loaded. We can do an exploration. So we can say head phenodata. So we have some of the rows being displayed. So we have the IDs, we have 2 columns also here for different groups. We have the sex group and then the population. And so for the IDs, the samples here, this column here will let us know which group they belong to. I mean the sex, so male and then female. And then for population, we have this also here to help us get that information. For this tutorial, our focus is to identify differentially expressed genes between males and females. And so our column of interest is the sex one. Let's proceed. So we can check, let's say dim phenodata to give us, let's check here, to give us the sample, the number of samples. So we have that also here. There are other ways to check the row numbers, but we will proceed with the rest of the activity. Now it's time to load the expression data using BALLGOWN. We already have it in the abundance directory, so we will load it here. So we say bg underscore chr x underscore, no underscore, so we have this, that's for the assignment operation or the assignment operator for R. So we have it this way, and we say BALLGOWN, and we specify the data there where that particular expression data is located. So it's located here in the abundance directory. And then comma, and then we say sample pattern equals ERR. And we have P data equals pheno data. And so the sample pattern here is used to get the information about the sample. So that means that it will look for this pattern, and all folders or directories that has this pattern will be loaded. That is what we are doing here. So this can also be a way to maybe filter out or to exclude certain samples. And then you also have to add the phenotypes, the information, and that is what the P data here is for. So we have already loaded that information here. And so it is this variable that we specify here. So once we do this, BALLGOWN will do the mappings and then gets all the information for us. So let's proceed. So let's load the expression data now. Perfect. So it's been loaded here for us. Let's clear the screen and continue. We can check some information about the loaded data, I mean the expression data, the abundances. So we can say class bg underscore chrx, and you have it here. So it's BALLGOWN. Okay, and we have all these things there. Now let's proceed. So you may have realized that I have been using the chrx a lot, and that is what was also being used in the original tutorial. This is because we are dealing with the human transcriptome, but the data has been downsampled, so it's only the chromosome X sample that are being used. Okay, so it's just a way to help beginners to try their hands or get their hands dirty with RNA Seq analysis using an ordinary PC or an ordinary laptop. Okay, so the human transcriptome, the entire transcriptome is huge, and so usually we use high-end servers or PCs to do the analysis. And so downsampling and giving us just a portion of it is useful so that we can reproduce the entire tutorial and commands using our normal laptops. So that is why we have this there. So it just tells us that we are using just the chromosome X data. Now let's proceed. So let's do some queries. We can say bg_chrx just to give us some description. So we have 3971 transcripts. And remember, we also used a command previously to get the total number of transcripts. And so Ballgown has also confirmed that for us here. Now let's proceed. So we need to also check some description here. Let's look at this command here. Methods class equals Ballgown. So this tells us what operations we can perform, okay, with Ballgown. So we have these methods here. So these are commands we can issue, okay, using Ballgown. So that means under the Ballgown library, we can issue all these commands and they will perform some operation for us. We are going to look at some of them, but the rest you may have to explore yourself. So let's proceed. So I'll try and then also introduce certain things to beginners especially so that you get a hang of the R syntax and then how some of the libraries are used. So just take note of that. So we are moving smoothly and I will say good job to everyone who is also practicing. So let's continue. Now with Ballgown, we can look at expression levels for different components. So you can look at expression levels for transcripts, for genes, and then for exons. I think exons is also there, and then I think we have introns as well. So depending on your question, depending on your research questions, you need to look at what you want to do. But for this exercise, we will focus on gene expressions and then transcript expressions. So we'll look at the gene expr and then the t expr. Yeah, they are here somewhere. Yeah, we have this and then we have this. Okay, so let's proceed. So that's what we are going to do. So to get the transcript expression levels, we say TEXPR and then we specify the Ballgown objects which we have available. We have it this way. And then once you do it, it will give you the gene expression levels here. Okay, so that's what you need to know. But because there are lots, that's why you see the rules just coming on and on. Remember, we had 3,971. So that is for transcripts. Let's proceed. So if you are interested in the genes themselves, then you can say gene expr, and then we have it this way. And then you run it. So that also give you the gene expression levels. And so you can look at the transcripts or you can look at the genes or other information. So just know what you want and then use the appropriate commands for it. You can also look at some few rows. Let's continue. If let's say you just want to look at maybe the first few rows for let's say the transcripts expression levels, you can use the head command again. So head, and then you give your TEXPR, and then you say BGX, and then you add your numbers. So it will give you, I think these are the first two rows. You can do the same with the genes. So this time you change this one to G, lowercase, and then you have that information as well. So these are some tricks you can use. Okay, now let's clear the screen and continue. It is also important to filter out transcripts with low variance. Okay, transcripts or genes with low variance. Okay, so it's something we normally do so that we'll be able to get results that have a higher statistical power. So it's important to do that. And so we are going to do some filtering and then remove all transcripts with low variance. And so this is how we do it. We are going to filter and remove transcripts with a variance 1 or less. And so we say bg_chr x underscore filt for filtered, and then we issue the command. So we say subsets because we are subsetting, and then we want only those with variance greater than 1. And so subsets, and we specify our Balgown objects, the original one, which is the unfiltered one. And then we also give an expression here. So row vars, and then we have texpr here, and I say bg underscore. So we have it this way, greater than 1. Okay, so that is the expression we are using here. And then we say genome subsets equals true, because we want to do the subsets. So that is what we do here to do the filtering. Okay, so let's run this command. Let's first check that everything is okay before we proceed. So I'm just trying to see that we have the right commands here. Okay, so I think it's okay now, so we can run it. Okay, so it's done. And so we can say bg underscore chrx underscore filt, and then we get what we want. So you see that the number of transcripts has reduced. It was from 3971, now we have 2746. So we have removed all those transcripts with variance 1 and then less. So that's what we have now. So we are ready to go. So let's proceed. So it's now time to identify the differentially expressed genes. And so let's proceed with that. So now we are going to identify the differentially expressed genes and then transcripts. And so to do that, we use the start test method in Balgown. And so for this exercise, we are focusing on transcripts and then genes. So we will do both here. And so let's start with the transcripts. And so we say DE transcripts. So that means we are looking at differentially expressed transcripts. And then we have our assignment operator here. And we say start test. And then we specify the filtered Balgown objects. So bg underscore chrx, and then filts. And then we say feature equals transcripts. And then we say covariates. So we have it here. It is equals to 6. And so what we are doing is that we are interested in identifying differentially expressed transcripts between males and females, and that is why we have covariates here, so that Balgown knows what to use. We also need to specify adjust vars, which is here. That's equals to C population. Let me just make sure I have spelled the population correct. So populacoi. Yeah, okay, it's here. population. Yeah, perfect. So that is it. So why do we have this? Aside sex, we can also have differentially expressed transcripts in populations. We are dealing with 2 different populations, and so that is something that can happen. And so this situation here, we call it confounding factors. And so we need to account for this, and that is why we label the population column as adjust vars, because we can have differentially expressed transcripts in this population, but that is not what we want. We are interested in the sex group. And so once we have this information, Balgown will know what statistical methods to use to account for that. And so it's important to always look out for confounding factors. Okay, so that is it for the transcripts. Now, let's add some other ones. So we have get FC. That means we want to get the full changes. So we say equals true, because some analysis also use this fold changes values, so we need to get that. And the measurement, we are using the ftp.ccb.jhu.edu. So this is a normalization method. So we have different measurements in that are given by STRINGTIE and then BALLGOWN as well. And so what we are interested in is to use the ftp values. So that's what we have here. And I repeat, there are several measurements in BALLGOWN, but this is what we want to use. So we have it done. Let's just check to make sure that everything is okay here. Okay, so I think we are good to go. Yeah, so let's run this command. Okay, so we have done that for the transcripts. And so we can just say dim, then that dimension to just get some information here. Yeah, so we have 2746. I mean, that's what we had when we did the filtering. So we can say head de transcripts to just look at the outputs. Okay, so we have that here. So we have the feature, which is transcript. We have IDs, we have fold changes, we have p-values, we have q values. Now you also need to understand that q value here, okay, is important because with RNA Seq experiments, we normally measure thousands of variables, okay, in small sample sets, and therefore this particular imbalance here needs to be accounted for, and that is where the Q value comes in. So Q value is also known as adjusted p-value or FDR adjusted p-value. Okay, so you need to always look out for this as well. Now for details about Q value, if you are interested, I will leave some useful links for you to read more about this. So basically, we do multiple testing to get this Q value, so just take note of that. And it can also be used as a criteria to filter out some transcripts. I mean, so those are things we'll look at later on. So let's proceed. So now we are done with the transcripts. So let's test on the genes as well. Now we are going to identify differentially expressed genes. So let's proceed. So we say DE genes. We still use the same start tests. And we say B gene CHRX underscore filts. And this time the feature is gene. Covariate is the same. 6. Adjust VAS is equals to population, the same as the other one. And then get FC equals true. And then measurements equals FPKM. We have it this way. So let's run this to also get the differentially expressed genes. Perfect. That is also done. So we can just do a dimension to get the number of rows and columns. So DE underscore genes. That is what the DIM command will do for us. So you have 921 genes identified. We have columns which are 5. Okay, the same as this because we are getting the same output. So we can even say head DE genes. Perfect. So we have it here. So here the features are genes. We have IDs, fold changes, p-value, q-value here. So that is it. So that is what we have here. Okay. Now you also observe that for the transcripts, we have just the transcripts, but we don't have gene names. It's important to also get the gene name because we need to look at which gene has those isoforms. Okay, so we need to have that label also here. It's important to get that information. Let me just put it that way because maybe you might want to do referrals, or maybe you might want to look at whether particular transcripts are isoforms of a particular gene. I mean, we need to look at all these things. So we need to add that identifier. Okay, so it's important to do that. We also get the gene names for this particular table as well. So that is what we are going to do next. So let's clear the screen. I'll start with a transcript. So let me first display the first few rows. So I'll say head DE transcripts. Okay. We are now going to add the identifiers. And so what the command we will do is that the command will check the transcripts we have in our data and then also look them up in the original BALLGOWN objects and identify the corresponding gene names. And then a new column will be added here so that we have the gene names themselves that we identified and then the corresponding transcripts, which is also here. And so let's look at how we do it. Once we have it done, you will see what I'm talking about. So let's proceed. The command that I'm going to use is also available in the original paper, so you can check that out. So let's do it. So we say de transcripts equals data.frame, and then we have gene names equals BALLGOWN. Gene names BAG CHRX underscore false, because we are doing a lookup in the original BALLGOWN objects. So we have it this way. So we are done with the gene names. Let's look at the gene IDs. Gene IDs equals BALLGOWN. We have our columns and we have gene IDs. We specify the BALLGOWN objects again. And then we add our DE transcript objects like this. And then we run this command. Perfect. So we have added identifiers. So let's display the first few rows again. So we say head DE transcripts. Perfect. So now we have the information we need. So we have the gene names, we have the gene IDs, we have the feature ID, FC, and all that. So we have the identifiers. So we will proceed. Let's also do it for the genes. Now we are done with the transcripts. So let's look at how we do it for the genes as well. So let's proceed. So let's clear the screen. Let's do a head DE genes. Perfect. So again, we are going to iterate through all the features here and also look them up in the original BALLGOWN objects and then get the names. So that's what we are going to do. So here we will do a bit of programming here, just a slight loop here. So it's going to be fun. So let's proceed. So first of all, we need to get the gene names. And so that's what we are going to do. So we are going to say bg first table. And then we say TEXPR. And then we say bg CHRX first again. And then all. We want all the columns. So that's what we have here. Okay, so we have that table. So after that, we are interested in columns 9 and 10. And so we say gene names equals unique. We want to remove all duplicates. So we have bg first table. We want all rows and then we want columns 9 and 10 like this. That is what we want. So we have that also there. Let's do a head. Let's say 2, just to get the first 2 rows. Perfect. So we have 40. So we have the gene ID, we have the gene name. Okay. When we look at the original DE genes table, you see we have the ID here. Okay, so what we are going to do is to look up this table here, look at the gene IDs, and then get the corresponding gene name. Of course, some of them don't have gene names, and so we are going to have some conditions here so that the program will know what to look for. And so basically, those that do not have the gene name will have a dot. So those ones, we will still maintain the dot for them. So anyway, let's look at how we will do this in R. So let's proceed. So we have the gene names. Now we are also going to get the features. Okay, that is the IDs. Okay, so we want to get the IDs from the DE gene table. And so we say features equals DE genes $ID. Okay, so we are looking at a column here, and that is why the $ID is here. So let's get the features. We are also going to create an empty vector. Okay, so that will be a container that we will store all the hits, all the identifiers that we get. So we say mapped gene names equals vector. And now we are going to issue some loops here. And so we say for I in features, what we did here, that is these ones here. And then we say, because it's a loop statement, we have to issue this. And then we say query equals gene names. Let's just have it here. Percentage here. Filter. So that's what we have here. And then we filter. So filtering, we say gene ID equals I. And we also want to get what? The gene name. Not equals to dots. That is what we have here. After that, we bring this semicolon here. So this is used to separate lines. So that is why I brought it here. So I'll say n hits is equals to then query. And then we want the first value. We issue another semicolon here. And we say if n hits equals 1, that means there was a hit. And therefore, if there's a hit, then mapped gene names equals append. We add the gene name itself. So mapped gene names. And that will be what? The query that we had, $gene name. And we have it here, like this. And then we close it. And then we also have this curly bracket to close that particular block statements. And we say else mapped gene names equals append mapped gene names dot here, and then we have it done. So that's what we are. So what we are doing is that basically we take each ID here and then we check the information here, because we can have one gene with which has 2 or more isoforms. We can have a hit where the gene name is there and there's also a dot. So what we are doing is to get the gene name. So if we are looking at this one here, for example, we look at it here and then we take the row that has this ID and also has the name not equal to a dot. We want a proper name here. And so once we have it, we add it to our empty vector here. But if we don't have any results, then that means that we will put a dot here. So that is what this command here is doing for us. So let's run this command to get our vector. Okay, so it has been done for us. So what we are going to do next is that this vector here that we have, what we have obtained, we are now going to add it to this particular DE genes table. So we will get a new column. And so we say DE genes $gene name. And then we say mapped gene names. That is what we have here. Perfect. And so what we are going to do next is to just look at the first few rows. So you can see head DE genes. And so you see that a new column has been added here. Okay, so that's what we have here. But if you also check, we may want to try and then organize our data better. And therefore, we are going to rearrange the table. Okay, so we are going to rearrange the columns here. Let's clear the screen and just look at it better here. Let's do a head again. And so we are going to rearrange the table. Okay, so we will rearrange so that the gene name comes somewhere here. We just want to get that here so that at least they are together. And so we will say DE genes again. And we say DE genes. And then we have C feature, the gene name. So we just want to arrange this the way we want to see it. So basically that is what this command is doing for us. Perfect. So we have it here. Let's close this. So let's run this command. But check and make sure that you have the same command as I have. Perfect. So that is also done. So now we can confirm by saying names, DE genes. So this will give us the columns. So we have feature, gene name, ID, FC, and all that. So we have it there. Okay, so that's what we have. So we can say head DE genes, and we have that also here. Okay, so now everything is in order, and that's perfect. So that is what we want here. And so this is very interesting. I believe you are also following and enjoying whatever we are doing now. So this is the world of programming, and these things are very interesting to do. Okay, so let's proceed. Now let's clear the screen and then just continue. The next thing we are going to do is to compute the log2 for changes. Let me first display the first two rows for one of the tables. Let's say DE transcripts. Perfect. For each of the tables, there is a column for 4 changes. That is the FC column here. Some analysis and visualizations require the log2 for changes. And that is why we are going to do the calculation. And then after doing the calculation, you create another column for each of the tables, and that column will contain the values. It will contain the log2 for changes. So that is what we are going to do here. So let's start with the transcripts. So we say DE transcripts, and then we create that column called log2fc. And then that column will have the values for the log2 for changes. So log2, and then we say DE transcripts. We specify the column on which this operation is going to be performed. And then we close our brackets. So let's run this command. Perfect, it's done. We can confirm by saying head DE transcripts. Let's use 2 to get the first 2 rows. And so we have the log2fc here. Let's also do for the DE gens. So we say DE gens, we have our new column, which is log2fc, and then we assign the operation to it. So we say log2 DE gens, we specify the column of interests, we close our brackets. Let's run this. Perfect, that's also done. Let's confirm. So let's say head DE gens 2. Perfect, it's also done. Now we are going to rearrange the columns here. We want to make our tables more organized. So we want the log2fc to join the fc here. So after fc, we want the log2fc to follow. So let's do it. So we are going to rearrange. So let's start with the DE transcripts table. So we say DE transcripts, and we say DE transcripts. We bring our square brackets, and then we have our columns here. We arrange it the way we want it to appear. So we have it this way: feature, ID, FC, log2fc, and then we have our p value. It should be in quotes, p value, and then q value. And then we close our brackets, and then we run this command. Perfect. So now if you do a column names, you will see that the rearrangement has been done. Perfect. Let's do same for the DE gens table. So we have DE gens, then we have it this way. So we just type the names as we want it to appear. So we have our feature, we have our gene name, we have our ID, FC, log2fc, p-val, q-val. Perfect. So now we close the brackets and then we run it. Let's just make sure we have done the right thing here. Okay, so it's fine now. Let's run this command. Perfect. So let's do a column names, DE gens, and that is also done nicely for us. So it's perfect. We are ready to roll. So let's proceed. Let's clear the screen and then continue. Okay, so let's take a look at one of the tables. Let's say transcript. So let's just call it again. Let's use first 4 rows. Okay. So what we are going to do next is to rearrange this particular table here again. This time we are rearranging the rows. We want to arrange from the smallest p-value to the largest. And so that's what we are going to do here. So let's start with the transcript. So we will say DE transcripts equals arrange DE transcripts. We are arranging using the p-value column. So we specify it here. Perfect. Let's do for the gens. So DE gens equals arrange DE gens p value again. Perfect. That is also done. So now we can do a head again. Let's just say DE transcript again. Let's say the first 4. Perfect. So we see it here. So you can see that there has been a change here. We are arranging from smallest to the largest. So that's how it is. Okay, so let's continue. Let's just clear the screen and continue. let's just call it again. Let's say 2. Okay. The next thing we are going to do is to save the results we have. So what we have here is the unfiltered table, okay? And so it's important to save this to an output file so that you can use spreadsheet software or any other tool to open them outside the terminal. So we are going to save the results we have now. And so what we are going to do is to write. So we say write.csv. We are going to write them to an output file. So let's start with the transcript. So we say de transcripts, and then we specify an output file. So let's say de transcripts unfiltered. Let's put it this way. So yeah, unfiltered.csv. We have to put in quotes, so it can be single or double quotes. So de transcripts unfiltered, and then we say row.names equals false, so that we don't have those numbers. These ones here, we don't want them appearing, so we use false so that R does not include them. So let's write the de transcripts table. Perfect. Let's also write for the degens. So we say write.csv degens, and then we say degens underscore unfiltered .csv, and then row dot names. Let's correct it here. Row dot names equals false. So let's check to make sure that everything is in order. So degens, perfect. So everything is okay. So let's run this command as well. Perfect. That's also done. And so we have that written nicely for us. So it's time to look at other stuffs as well. Let's continue. Let's clear the screen and then continue. Filter. The next thing we are going to do is to check how many transcripts are significantly expressed. Now we know there are transcripts and then genes that are differentially expressed, but then we need to look at those that are significantly expressed, okay, statistically speaking, okay, so we need to look at that. So whenever we are looking at some analysis, we need to look at those that have significant values, and so we are going to do that. So we are going to do some queries and then filter and then get those ones that are significantly expressed, statistically speaking. And so let's start. So let's proceed. So let's check. We will use the adjusted p-value for this. And let's just first do a head. Let me start with the transcripts. Perfect. Let's have it this way. So let me say that in identifying those that are significantly expressed, you need to look at threshold we will use. Some will use Q values, some will combine Q values and then P values. So this particular threshold, you need to define it yourself. You need to decide it. It's very important. And so depend on your research questions and then depend on what you have, then you come up with a threshold that we'll use. You can also check papers and publications and look at what thresholds have been used and then adapt them. And you also need to consider the organisms you are using, sample size and all that. So that's one. There wouldn't be a fixed threshold, that's what I'll say, but there are best practices. And so I will encourage you to read papers, check publications, and look at what is being used and then try and then adapt that. So that's what you need to do. So again, I repeat, the threshold we are going to use here, may not be applicable in all situations, but it's just a guide for you to use. So let's proceed. So we are going to check how many transcripts and genes are significantly expressed, and we are going to use a Q value threshold of less than 0.05. So let's start. So first of all, we are going to count. Okay, we are going to do a count. So we say table the transcripts $Q value, Q val. and we say less than 0.05. We have it this way. So this will give us just the count. Perfect. So we have false and true. So false here refers to transcripts that do not meet the criteria. Transcripts where the KO values are not less than 0.05. Those that have their respective KO values less than 0.05, what we consider as significantly expressed in this tutorial, are 10. So that is what we have here. And so this command here will just give you the numbers. Okay. If you want to check the indexes, the particular rows where the hits are found, these ones are found, you can use which. Let me just do it again. I'm also using this to show you some tricks that you can use in R when you are analyzing data. You can say which, and we say D transcripts QVAL less than 0.05. Like this. Perfect. And that will give you the indexes. Okay, so that's what you need to know here. So these are tricks you can use. You can also repeat for the gens. Let's just repeat for the gens. Let's do it as well. So you can just do that and then just check what you have there. Okay, so let's clear the screen and then just continue. So I'll just repeat again here. Again, so let me just repeat so that you see how it goes. So we say for transcripts, I will say table D transcripts QVAL less than 0.05. Perfect. So I have 10 of them. Let's do same for the others. Now, what we have here is giving you the counts, but if you want to get the list itself, then what you have to do is to say D transcripts, and then this time we filter, and we say QVAL less than 0.05. So this will give you the hits. Okay, so we have them here there. Yeah, so we have 10 of them, just as the count said, and that's what we have here. So we have 10 of them here. This one is here because there's another column, but because it cannot fit this space, that is why it is here. But basically we have all of them here. So we have the transcripts, we have their IDs, and then the gene names as well here. So that's cool. Now let's proceed. So let's do same for the genes as well. So let's clear the screen and continue. So we are going to do same for the genes. So we say table D genes $QVAL less than 0.05. Yes, so for genes there are 7. So that's what you need to know. Let's check the list itself. So we say D genes, and then we say what? We filter, and we say QVAL less than 0.05, like this. Perfect. So that is also done nicely for us. So we also have them here. So that's it. What we did was to display them, but we can also subsets so that we have them in a different table. So let's create 2 different tables and then put those transcripts and genes that are significantly expressed in those tables. So we are going to have 2 tables. Let's start with the transcripts. So with the transcripts, we will say, let me just check again. Perfect. So with the transcripts, let me just clear the screen and continue. Yeah, so with the transcripts, we are going to use the subset command again, and then the genes too, we are going to use subset. So the subset function allows you to subset data frames and tables. That's what I would say. So it's important to have this at the back of your mind. So we say subset. I'm starting with the transcript. So I'll say subset underscore transcripts. And then I'll say subset again, and I'll say D transcripts. Okay, this is our table of interest, and I'll say where D transcripts has the column, which is kill value less than 0.05. That is what this command basically means. So let's run this command. Perfect, it's done. So again, you can just say your head subsets, let's say 2 or 1. Let's just say 1 to get it then. And then you have it done. Okay, so that's it. Let's also do same for the gens. So you say subset gens, and I'll say subset again, D gens where the kill value, that's the kill value, is less than 0.05. That's it. And then I'll close the brackets like this, and then I'll run this command. Perfect. Again, I can say head subsets gens, let's say first 2. Perfect, that's also done nicely for us. And so what we can do is to also save these to output files. And so we can say, let's start with the transcript. Write.csv subset transcripts, and then we give the output name, which is D transcripts underscore filtered. You can use any name of your choice as well, CSV. And then I'll say row.names equals false. Let me also save the D gens as well. The subset. So I'll say write.csv, and then I'll say subsets gens, and then D gens filtered. That's the output name, csv.csv, and then row.names equals false. Perfect. That is also done nicely for us. So this is how we identify differentially expressed gens and transcripts using some R packages. Now let me show you how you can also extract the read counts and then save them to an output file. We are going to extract the read count for both the transcripts and then the gens. So let's start with the transcripts. So we say fpkm. We are extracting the fpkm, by the way. And then let me say underscore transcripts. We want that for transcripts. So this variable here will contain that information. So we say texpr, and then we specify the Balgown objects, the filtered one. Now I also want to get additional information. So let me just say I want all the information in that particular object for the transcript. So I will say all like this, and then I will just execute this command. So now I have them. So let's look at the column names. I'll just use this one here, names fpkm underscore transcripts, to get the columns. Perfect. So these are the columns. So we have the fpkm values, and we also have some coverage information and some other ones here, transcript ID, etc. So we have that information. So we are not going to exploit it, by the way. You can exploit it also. So I'm going to show you how you can also save this particular information to an output file. So let's first clear the screen and continue. So I'll say write.csv, and then I will specify that particular data. So that's what fpkm underscore transcripts, and then I'll give the output name. So let me say fpkm underscore transcripts. This time, because it's an output file, we need to put it in quotes. So underscore transcripts.csv. And then I'll say row.names equals true. This time I'm saving everything. So that's how we do it for the transcripts. Let's do that for the gens as well. So for gens, I'll say ftp underscore gens. And then I'll say gexpr. I specify the BALLGOWN object again. The filtered one. Like this. And then I run it. So here, let's check the names. ftp underscore gens. Let's check. Okay, so here it's not because it has a different data type. So what I can do is to just say head ftp gens to get some of the information here. So that's fine. Okay, so the null here is because probably the data type here, I cannot use this particular command to get the column. So that is why it gave null, but it's fine. Don't worry. We are good to go. Okay, so let's proceed. So once I have this information, I'll also save it. So let me just clear the screen and continue. So I'll say write.csv, and then I'll say ftp gens like this, and then I'll specify the name here, ftp gens.csv. row.names equals true. I'll save everything here. Perfect. So that is done. Okay, so now we can exit R because we are done with the analysis. And so let's quit R. So to quit R, you say kill, and then you give your parentheses. And then you'll be asked to save the workspace. But this time I'll say no because I don't need to do that. And then I'll exit. So now I have exited the R interface. So let's do an ls. And we are going to see the CSV files, the one that we saved. They are all here. Okay, so we have them here. Okay, so let's look at what we are going to do next. Now we can also organize our files better by putting the results, everything that we generated, into a different directory. Okay, so we are going to do that. So we can say make the, and we say results. Let's say results. And then we can move everything there. So I'll say mv star.csv. I'll move everything there. Okay, so now if you do an ls in results, you will see all the files that we generated there. So let's proceed. So now let's clear the screen and continue. Let's clear the screen and do an ls again. Perfect. So now we are done with the first stage. Now it's time to generate some visualizations, because as the saying goes, a picture is worth 1,000 words, and sometimes it becomes easier to interpret and understand the data if we generate plots and then we are able to study them as well. So there's this R script here that we'll use to generate some cool plots here that I am going to explain later on. So let's do that. So for this particular script, all that we are going to do is to execute the scripts. So let's do that. So let me just see whether I can show some of the R scripts here before we proceed. So the scripts can be opened using your RStudio. So you can do that and then just read through it. But yeah, so all the plots are here. So there are also comments there to help you understand them. So that's it. Now let's go back and then run the scripts and then get the visualizations done for us. So to run an R script on Bash, we say R scripts, and then we specify the R script here. So let's do it. So let's run this to get the plots generated. Okay, perfect. So the plots have been generated. Let's clear the screen and continue. So let's do an ls, and you will see a new directory called plots here. Okay, so this plots directory contains the figures that have been generated. So do an ls into it, and we are going to see the figures there. So that's how it is. Now let's move this plot also into this results directory. So I'll say mvin-vin plots, and then I'll move them to the results directory. Perfect. So that is how we do it. So now let's go to the GUI. I'm just going to show the results on my PC for you to also look at them. Okay, so we have the files here. You can open them. The CSV files here can be opened using any spreadsheet software. So I'll just open them here. So this is how they look like. I will just enlarge the text here. So let's start with the transcripts. So these are the transcripts. So we have the IDs, the fold changes, log2 fold changes, p-value, p-value. So here you can manually inspect the data you have here, and then maybe try to get some additional information here. We have the filtered one here, which you can also check. There's also the FPKMs we generated for transcripts. So here we have the IDs, the genes, the FPKM values themselves, etc. So you can just manually inspect and then just look at what you need. You also have the information for the genes. They are all here. The unfiltered, the filtered, and then the FPKM values, they are all here. So this is how you extract the information and also try to manually inspect them. Now the plots are also here in this directory called plots. So here we have some plots here that I'm going to explain very soon. So we have about 5 files here. Yes, so there are 5 figures here. So you should have the same. So let's go back. Now let's go to the /u, and then I'll explain these plots in details. So let's look at the first figure, which shows the structure and expression levels of isoforms for the gene SIST in sample ER188234. If you look at the figure here, there are 6 isoforms for the gene, and the expression levels for these isoforms are represented using a color scheme. So the color scheme ranges from this side here to this side. So it's yellow, by the way. And so the lighter side here indicates low expression, and the darker side here represents high expression. So if you look at the 6 isoforms here, isoform number 4 is highly expressed compared to the others. Okay, so that is one cool thing about the pipeline we are using. It allows you to study in detail a gene as well as the expression levels for its isoforms in a particular sample. And so that is how it goes. Now let's look at the next figure, which is the volcano plots. Volcano plots are 2D scatter plots that have the shape of a volcano. With volcano plots, the log2 fold changes is on the x-axis, and then on the y-axis we have the negative log of the p-values. And by setting a threshold, you'll be able to identify genes that have been expressed significantly. So that means we are looking at significantly expressed genes. That's what you can use the volcano plots to do. And that means you'll be able to go further and identify down-regulated, up-regulated, or those genes that are not differentially expressed. So this is how volcano plots can be used to visualize the data. And you can represent each of these groups using a color code here, as has been shown here. With the volcano plots, you see that the left side has the down-regulated genes, and on the right side we have the up-regulated genes. The middle side here has those genes that are not differentially expressed. You can go further to give additional labels to genes that are of interest to you. I made mention of threshold. So let's look at the R code so that I'll show you the threshold that I set to identify down-regulated and then up-regulated genes. So the threshold is here, it's in the code. So genes that are up-regulated should have their log2 fold changes greater than 1, and also the p-values less than 0.05. And then those that are down-regulated should have log2 fold changes less than -1, and then the p-values less than 0.05. So that is how it is. Again, there is no standard threshold, and so you will have to decide what threshold works best for you. And so I advise that you look at literature, you consult the experts, and then use whatever information you get to help you to set an appropriate threshold. So just take note of that. Let's go back to the plots. So using this color scheme, it becomes easy to pinpoint these genes that are of interest and then do the interpretation better. Let's look at the next figure, which is the MA plots. MA plots are also 2D scatter plots. They are used to visualize gene expression data and also identify changes in gene expressions between two conditions. This plot here represents the transcripts. With MA plots, you have the log2 of the means of the normalized read counts on the x-axis, and then on the y-axis we have the log2 fold changes. And by setting a threshold, you'll be able to identify those genes or transcripts that have been significantly expressed. So that is what you need to know. For this particular plot, I use the KO value to set the threshold. So KO value less than 0.05 indicates differentially expressed, and then those that are not less than 0.05 means that they are not differentially expressed. So that is how this can be interpreted. And we have a color code here to represent those two groups. With MA plots, the up and down regulated genes are at the extremes. So at the upper part here will be the up-regulated genes or transcripts. In our case, we are dealing with transcript. This plot is for transcript. So the upper region here will be for up-regulated transcripts, and then the lower region here will be for down-regulated transcripts. And in the middle here are those that are not differentially expressed. Okay, so using the data we have and then using the color schemes, you realize that all the transcripts are in this region. And so this means that these are down-regulated transcripts. I'm making this interpretation based on what I'm seeing here. So take note of that. So the transcripts here from the image here, they are down-regulated. So that is for the MA plots. Now let's look at the last plot, which is heat maps. Heat maps are a great way to visualize and interpret your gene expression data. And so with heat maps, you usually have a color code here to also help you to do the interpretation. This heat map was generated using the differentially expressed genes. So we have their expression levels across the 12 samples here. And so using the color scheme here and then comparing them to each other, we see that those genes that have low expressions have this color here. They are in this region, and those that have high expression levels are in this region. And so if you look at this heat map here, if you look at this figure, the gene MSTR gene.728 has been expressed in high levels across the samples, all the samples. This is followed by this, and we also have the others here. And so if you combine heat maps with clustering methods, you'll be able to identify groups of genes or transcripts that have similar expression patterns like what we have here. And so by using these clustering methods, you'll be able to identify genes that are commonly regulated, and this can also help you to identify biological signatures that are associated with a particular condition. So that's how you can use heat map to interpret and then also make some inferences. And so here you can see that if you look at the genes here, there are two main clusters. Okay, so we have cluster 1 here, which have high expression levels, and we have cluster 2 here. So comparatively, we have two groups, those that have been expressed in high levels, and then those that are, comparatively speaking, those that have low levels. Okay, so one is higher than the other. And so what we see here, that these two have similar expression patterns, and then this particular one here, this other group here also has similar expression patterns. And here you can also go further, okay, by using this clustering approach here to try and then get a deeper understanding of your data. The same can be used for the samples as well. So heat maps are a great way to try to interpret your gene expression data. So I encourage you that you also use heat maps to do the same with your respective data. In addition to these plots that I have just displayed and also explain. There are other visualizations that you can do to understand and interpret your gene expression data, but these are things that we will cover in later tutorials. And let me also say that this tutorial was made possible because of the works of others. I had to read a number of articles and tutorials, and then I took quotes from these materials to prepare this tutorial. So I want to acknowledge all the authors and the creators of those materials. So I'll just show them here. So this is the original article, and then I also got some information from Dave Tang's blog, and then we also had some information from the Canada Bioinformatics group. I will leave all the links in the description box. There's also this particular one here, Pi DuPont, and then a host of others. So I'm going to leave all these links in the description box. And I also want to thank all these authors again for the work that they put in into getting all this material. So I want to acknowledge them again for a good job done. So let's go back. Once you are done with the analysis, what you need to do is to deactivate your environment so that you can just close it. So I'll just say conda deactivates. Perfect. So now I have deactivated the environment, so everything is now clear. Now I also want to thank you, my viewers, who had time to watch this tutorial, and you can also give your comments, and then we can have some discussions going. If there are things that you think should have been added, you can let me know, and then in the future tutorials I'll try to include them. If you also want to support this channel, there's a buy me a coffee link there that you can just use to buy me a coffee, and then you can keep the spirits going. So that'll be all for this tutorial. Thanks for watching, and I will see you in the next session. Goodbye.